Well, welcome everybody to Apiculture Online Hive Chat with NC State. I'm David Tarpey, NC State Professor of Apiculture, and joined here by our usual cadre of folks from NC State, as well as our Apiary Inspection Service and the NCDA. And welcome to all of you who are on YouTube. We have a really excellent lineup tonight talking about different aspects of bees and beekeeping, and a very special guest all the way from the University of Minnesota, Dr. Katie Lee, who's we're going to be talking with later on in the segment, the third segment. But for now, for all of those of you that are joining us on YouTube, please use the chat function to ask any questions. We'll try to get to as many of those as we can in the fourth and final segment of our Q&A. And also be sure to ask any questions of Katie and others as we go through the evening. But also be sure to like us and subscribe to our YouTube channel so you get automatic updates and notifications of whenever we're doing any of our live Apiculture Onlines. But for now, what we're going to do is we're going to start with the first segment that we always start with, and that is our bees in season. Before I do, I want to make a very, very quick announcement to both thank all of you who have been sending us your drone laying queens and have seen this announcement either in former Apiculture Onlines or through other flyers that we've been putting out there. But Brad Metz within our program is, and with Jennifer, of course, is doing a field study on trying to recover drone laying queens. And so if you go through your colonies and you see something like this, where the brood in the middle of the brood nest, where the worker brood should be, but you see nothing or mostly domed out drone brood within the brood nest, we want that queen. So if you send her to us, we will analyze her and see if that is actually a recoverable condition. Most of the time, you just want to replace that queen and get rid of her anyway. So rather than get rid of her, go ahead and donate to science. So if you have any questions about that, email us here at the Honeybee Queen Clinic, all one word, at ncsu.edu. And thank you again to all of you who have already sent in queens. We've gotten a good handful of those so far, but as usual, when it comes to research, the more is usually better. So just let us know if you're seeing any of these things. So again, our first segment that we always start out with every single time in Apiculture Online is the bees in season. What your bees are doing right now and what you should be doing as a beekeeper to help and assist them so that they can continue to thrive and be healthy. And so, in late April here in North Carolina, within the region, spring has been kind of taking its time this year. Spring has certainly sprung. It sprung kind of a month or more ago. But it's not really taking off this year, at least in our neck of the woods. That might be different in other areas of the region. But it's kind of been more drawn out. And so that's both good and bad in the sense that the bees aren't booming. They're not going from 0 to 60 in a heartbeat and getting away from you. But that also means that swarming season hasn't really hit as hard and heavy as often is the case this time of year. But the tulip poplar and the other main nectar flows are, starting and doing very well. Those of you on the call can confirm or refute that. But, right now, this is the real kind of prime time where the main nectar flows are underway. And so the bees are busy out there getting all of the nectar in abundance. So what that really means for us as beekeepers is you just want to super up and let the good times roll. You just want to kind of hopefully the things that you've done in your early spring management or colony establishment, let the bees kind of build up and just do their thing. This is kind of the downhill sledding portion of the season when there's lots of food coming in, the bees are growing and hopefully doing well. And so that's something that you just want to capitalize on and stay ahead of them, especially when it comes to colony growth. So a lot of you are also installing new colonies from packages, either if you've never had bees before and you're starting new colonies, or you're growing your number of colonies and not using splits, or you're making up for any winter losses that you may have had this past year. You know, the packaged bees is just one of the real familiar starter kits of starting a new colony. And so I just want to go over, for those of you that have already done it, or for those of you that are about to get your packages either for the first time or first time this year, just remember that you know, shaking them out during the middle of the day, just like Jennifer's doing here, right, into a brand new colony, shaking them out like that isn't necessarily the best way to do it or the only way to do it. You don't need to shake during the middle of the day, because when you do this, especially when you do this to, you know, multiple colonies at the same time, a lot of the bees, the older bees, they take flight. And so they're flying around, not in any sort of defensive way, but they're flying around out of confusion, and, you know, things are going crazy, and that can cause a lot of drifting. So, you know, we see this oftentimes where, you know, you shake 10 packages into 10 boxes, but all of the ones that are flying around, they all drift into one of them. So you have one hive that's just bursting at the seams with maybe 8 pounds of bees, and the other ones, you know, have a lot of attrition from them, and so they don't have a critical mass, and you can lose queens and colonies that way. So, you know, shaking them right in the middle of the day is not necessarily the best and only way to do it. And so if you're installing new packages this way, an alternative way to do it is rather just taking the queen out of the package and keeping her in the cage, putting her in the hive just like you would normally, maybe shaking a couple bees just to cluster around her. But then you put the inner cover on, and then you put an empty brood box, a shell brood box, and then you take that hole in the package and you place that upside down on the hole in the inner cover, and just let the bees walk out overnight. As they start to realize where the queen is, they'll just kind of naturally move down, so that they're not going to take flight. You're not going to have a lot of that confusion, drifting, and everything else. So, you know, that's an alternative way that you can do it at dusk. You can do it, you know, at any time, and just let the bees kind of naturally walk in. So that's one tip if you want to install packages kind of in a different way. Another tip, and this is actually based on some brand new research that just came out that was spearheaded by a master beekeeper here in North Carolina, Eric Talley, for his master craftsman research project where he collaborated with us. He set up colonies from packages using the standard method of just shaking them in, just like we described. But then there were other ways that they were established too. One was to establish the colony where there was a foil plate of brood pheromone that we created and placed into the colony. And then another treatment of packages where they were shaken into a colony with one frame of open brood, eggs and young larvae. And the idea here was to see how that affected the success and the establishment of these different packaged bees, because a lot of people experience problems when they're installing packages, like I explained. And so this is just one finding from his study that I think is really, really exciting, and something that we want to promote and let people know about. And that is, if you install packages just by shaking them out in the normal, typical way onto nothing but foundation, what we found was that about 60% of the queens in those cages, in the packaged bees, got superseded. They got replaced within the first, I think it was 12 weeks. And so that's obviously not very good, right? That causes a lot of downstream problems, cascading problems, where the bees get more stressed out when they don't have a laying queen. open themselves up to disease and other problems. And so, 40%, 4 out of 10 colonies that experience that is really not good. I should say that an additional 25% or so of those packages, they started building queen cells, but then they kind of didn't go through with it. So they tore down the queen cells. So they kind of thought about getting rid of the queen, but then they ended up not doing that. So those queens still survived, but they kind of came precariously close to getting replaced. And then about 25% of the colonies, they just never built any cells. They were good to go from the very beginning. So normal colony establishment, 60% of the queens survive, 40% of the queens don't. But if you contrast that with the packages where they installed into a hive with just a single frame of open brood, 90% of them, the queens were accepted, no problem. And only 10% of them, they started building queen cells, but then they reverted and they kept the queen anyway. So it was a much, much better percent acceptance if you install your packages into a hive that just simply has one single frame of open brood. Now, if you don't have any other hives, you're just starting from the beginning, you're not going to have that. Or if you can go around and ask your mentor if you can get a single frame of young larvae to install in your very first hive. But if you're installing packages, but you already have some that overwintered, this is a great way to take, one frame of bees, of larvae without the adhering adult bees onto them, placing that into your hive as you're installing the packages. It will greatly improve the success of the queen and therefore the colony getting established. So it's another new kind of tip that hopefully will really help improve the establishment of newly introduced packages. So another thing that's really important as a beekeeper and what you need to do with bees in season is you want to be looking ahead 1 to 2 brood cycles from now, which is 3 to 6 weeks from now, because you want to predict what your colony is doing so that the management things that you're doing to them right now will help set them up to where they're going, their trajectory, right? So right now, things should just continue to grow during the spring season, right? So you want to stay ahead to make sure that they don't get too congested within the hive. Make sure you put on additional hive bodies, give them honey supers for them to fill, all of those kind of things. They're going to continue to grow. You really want to stay on top of your queens and swarming. You don't want that brood nest congestion to occur so that they then go into swarm mode, because if they swarm and or replace their queen this time of year, again, that can have these cascading problems downstream where the colonies are just going to be more stressed out during, going into summer, which is not what you want at all. And then finally, this is always a recurring theme. If you haven't already, start checking for those mites. It's pretty much never too early to start checking for Varroa mites. Presumably those levels are going to be fairly low right now, coming out of winter or starting new colonies from packages, but you never know. The mites can go from 0 to 60 in a heartbeat. And if you have relatively high numbers now, they're going to be astronomical later in the year. So right now is the time to start checking to implement any sort of control measures that you might want to make sure that your bees are nice and healthy. And so with that, we'll then move over. I should say, are there any questions or comments, both from us here on the Zoom call or anybody online? Let's see if there's any questions. I don't see any on the YouTube channel, but anybody want to comment on anything on the bees in season?
I wanted to remind folks that it's really important to feed those packages, and that food needs to be really close to the cluster. I've responded to a couple of calls this week of dead packages, and they had a cluster of bees at the top and a boardman feeder in the front entrance. And during this cold weather this last weekend, that was a recipe for disaster.
And so why do they need that feed, especially when they're being installed onto foundation, Lewis?
Yeah, so they need that food to draw comb, and they need those calories to stay warm during this cold weather. And if they can't get to the food, if the food is not right on the cluster, it's lights out.
Exactly right. They convert all that sugar into the wax, which takes a lot of energy. So they need that. And smaller colonies, especially when they're established, they might not have the foraging force to be able to bring in enough nectar to do that. So yeah, definitely want to be feeding sugar syrup as you're installing those packages. Thanks, Louis. So with that, our next segment is our timely topic. And so for this evening, we're going to turn that over to our very own apiculture technician, Jennifer Keller, to talk about making splits, which is another way to start new colonies, and how to handle making divides.
All right, hang on, let me get my screen going. Okay, there we go. All right, well, thank you. And so, yes, I'm supposed to be talking about spring splits. Put that in there for some laughter. But really and truly, this is the time of year we see so many swarms. This year's not been as crazy as it was last year. So that's a good thing. But I think we might be a little delayed and swarm season is still upon us. But this is when our hives are building up and the population is really building quickly. And the hives can get overcrowded before you know it. You can go in there one day and everything looks great. And then you go in a week later and it's just, bees wall to wall and they're ready to swarm. So if nothing's done, then the bees will take off, which I think they get a bad rap. Swarm is not really all that bad. It's a sign that you have a very healthy colony of bees. So that's a good thing in one way. But the downside is that you end up losing half of those bees along with the queen. And if you're able to catch them, then that's a positive. You can start another colony. But lots of times they go way up in the trees and they're just gone. You don't want to risk anything to try to catch them. The remaining part of your colony will be fine. They'll requeen and you'll hardly be able to tell that you lost any bees because they'll replenish themselves so quickly with all the brood that they left behind. But the one issue is you'll lose time while you're waiting for that new queen to go out and get mated and come back and start laying. So there's a period of time where population won't be increasing anymore for a little while. And so that's what we're trying to we don't want that to happen. So instead of letting the bees swarm, why not make the swarm on your terms by splitting the colony before they actually leave? So it's like making a swarm on your own before they do it themselves. Now, I want you to notice the picture of that box to the right. That is a nice full box of bees. Okay, that's what we're talking about when we're talking about splitting a hive. So we're going to go a little bit further into this. So rules for splitting. Well, actually, there are no rules. There's no one right way, but there are plenty of wrong ways. And I was trying to think about all the different ways to make splits, and I was going to write some down, and the list got so long that I just quit. You can spend all day looking on YouTube at different methods of hives. But there's just a couple of things to keep in mind that are musts. And the first must is think about that picture that you just saw. You must start with a healthy populous colony of bees. Now, if you have a weak hive, and I've seen people try to split pitiful hive, they want more bees, they want 2 hives. But if you split a weak hive, you'll end up with 2 weak hives, and neither of which will be able to not only make it through the winter, but they might not even make it through the summer. If they can't feed themselves, they could run out of resources and just die before winter ever gets here. One of the biggest complaints I hear about people when they're trying to make splits is that they can't find the queen. So basically they just give up and don't make a split. So the good news is that it's not necessary to find the queen in order to make your split. it's helpful. And if you can find the queen, then it makes life a little bit easier, but it's definitely not necessary. And so you've heard the term walk-away splits, which is basically just taking half and put it into one box and the other half into a separate box. If you find a queen, you can pick and choose what frames you put in there a little more carefully. If you can't find the queen though, all you have to do is make sure you have some eggs and larvae in both halves. And as we know from our basic queen biology, that they're made from larvae that are less than 24 hours old, ideally. So that we want to make sure we have in both halves, so that whichever half doesn't have the queen will be able to make another one. And ideally, like I said, ideal situation is to have a second mated queen ready to introduce into the queenless unit. That would save you a lot of time, but it's definitely not necessary, since you're going to be putting resources into both boxes to make a new queen. So you hear the term walk-away split, and there's really no need to do too much work into figuring out what goes into each box, as long as you have some food in both. For example, I had to move some bees not too long ago to make room for a research project, and they were very heavy. So I didn't want to spend too much time because I didn't want bees flying all over the place. I just picked up the top box and put it on the bottom board and put an introducer on, closed it up, put a lid on the bottom box. So I now have 2 boxes of bees. I have no idea what's in either box, but I took them back to the lab, set them up, and then 4 days later, I went in and figured out which one had eggs, because, you know, after 3 days, all the eggs will be hatched. So if there's a queen laying eggs, then there will be eggs in the hive. The hive that didn't have the queen, I found queen cells. So at that point, it was very easy to determine which one had the queen. And there was like no work done whatsoever. And if I didn't find queen cells, I could have then gone ahead with figuring out how to requeen that half. It didn't have to be done right then. It's helpful to do it right then, but you have time. And this was just a means to make a split on my terms, not on the bee's terms. So, like I said, the ideal situation is to have that caged queen purchased or given to you, or like somehow you come up with an extra queen. I don't know how, but like maybe you win it at a door prize. You have an extra queen. So that would be the best case scenario. You put that into the queenless unit. And in this case, you would have to find the queen. You would have to make sure that you're placing her into a unit that did not already have a queen. Because if there was a queen in there, the worker bees may not accept her. They would probably just kill her before she ever got out of the cage. So that's the one instance where it is important to find the queen, or like I said, wait 4 days and be sure which half she's in. If you don't have a queen, there's other ways to speed up this process a little bit. The bottom right picture is a picture of queen cells. I think I stole this picture from Don Hopkins. This might be actually why you do a split. You may be just going through a normal inspection and you find a frame and you realize, oh no, this hive is getting ready to swarm. So I better make a split now. Well, you can take some of these queen cells and put it into the new unit that doesn't have a queen, or put some into each unit to make sure you have queen cells in there. And then you don't have to worry about them starting the process of making a new queen. They'll be way ahead of the process. The top picture is just how we make them in the lab. And so each one of those I can remove and put into a separate queenless unit. All right, and then one more. This is just what I did with 5 of those really crowded frames, put it into a 10-frame box, add some extra ones on both sides. I have an open brood, cap brood, and honey and pollen, whatever numbers works for you. Queen is optional. And in this case, I added 3 brand new foundation frames. This is a great time to get them to draw out some new comb for you. All right. And then I hear people say, well, I don't want any more hives. What do I do if my bees are getting overcrowded and I don't need any more hives? Which I just can't imagine anybody saying that, but it might happen. But this is a great opportunity to make up a nuke. This is a picture of a 5-frame nuke. I just took out 2 frames to get the picture. But this is you can sell these or donate them to other struggling beekeepers that may have just lost a hive, which I've read a lot about on listservs just recently. So that would be a great use of a split. And it's always a good idea to have an extra queen or 2 on hand. And this is a great way to house them so that if your 2-story hive that you're using as a honey producer, if you accidentally crush that queen during one inspection, you can just go in here and take this queen and introduce her. So it's just it's a fun way to play around with some queens as well. And if by some chance you don't use the queen and it makes it through the summer, and you can't overwinter nukes here in North Carolina. Not always, but they don't eat as much, so they're easier to baby through the winter as well. But that split in the spring, come February or March, they'll be ready to take off, and it'll be a great new colony for next season. So get out there and split your bees. That's all I have. Any questions?
That's great, Jen. This definitely is the height of the season for that. And I totally echo your sentiment about, having some reserve nukes and colonies on hand. You might not want, to grow to 100 hives or anything, but still having more than you think you need, because, things do go wrong. There's some questions here on the YouTube, and I'll kind of paraphrase a little bit. So when can one expect to get a honey crop from these different methods of creating new colonies? So talk about packages, but as well as these different kinds of splits. So what would you kind of estimate under normal circumstances?
So a package, you probably will not get a honey crop the first year. They're using all their resources to make the comb and build up. So, next year for a package. This 5-frame nuke, if you get a really good busting, you get 3 frames of capped brood in there, they'll be able, and you move that into a 10-frame box, they'll fill up the 10-frame box in a matter of weeks, and you'll be ready to put the second box up. So it is really beneficial if you can find a nuke. That's the problem half the time, is getting it early enough. But they build up so much faster that you can afford to wait a little bit longer to get a nuke, because I think I could probably get a honey crop off of a good 5-frame nuke this summer, possibly.
Yeah.
If I have all drawn comb, for example.
See, any other questions from the gallery or from YouTube here? You were also mentioning the walkaway splits as another means, or doing a dirty split where you don't necessarily know where the queen is, but you go back 3 days later, and whichever one has eggs, the other one is the one that needs a new queen, right? I think that's an easy thing. Are there alternatives to doing that, like shaking all the bees through an excluder or some of those other things? Like, would you recommend that dirty split over kind of a more heavy-handed way of finding the queen?
Well, if you are able to plan ahead, which we always try to do but often fail to do, but if you want to make a split over the weekend, if you put a queen excluder between your 2 boxes 4 days ahead of time, you can then easily go out and tell which half of the boxes she's in, and that will speed that part of it up. The downside to not knowing, or even just putting queen cells in, is that it possibly might not work, but you can always recover from a failed attempt. You can always combine them back together. Right. It may not work, but it's always worth the trying of it. I think it's more successful more than it's not. But you need to.
That's all great advice. Of course, getting used to finding and locating the queens, even if they're unmarked, is always a really good skill to develop for beekeeping. But sometimes that can take practice. But these are good tips to if you're not able to do that as readily as you might. So, but thank you again, Jen. That was really great. I think what I'm going to do is then go to our next segment here, which is our guest interview with our special guest of honor tonight, which is Katie Lee. Katie Lee is a postdoctoral researcher at the University of Minnesota, but she has worn many hats, not just this woolen one here, but she's worn many hats over the many years that she's been in the University of Minnesota. She started, I believe, as a master's student. Correct me if I'm wrong here, Katie, but started as a master's student with Marla Spivak. But then she became one of the very first tech transfer team members of the Bee Informed Partnership, both in Minnesota and out in California, and has moved around. After really doing a lot of great work with the BIP, she then decided to go back and got her PhD under Marla at the University of Minnesota, and has stayed on there then doing other research projects at the University of Minnesota. So she's really come a long way during her tenure there. And we're really, really lucky to have her because she's one of the most sought after speakers on the national circuit and has done a lot of really, really interesting work. So welcome to Apiculture Online this evening, Katie.
Thanks so much for having me.
So tell us a little bit about what you've been up to recently. Hopefully that was an accurate enough history and bio of what you've done, even though that was brief. But right now you're working a lot on not just honeybees, but also on native bees and how they interact in the environment. Is that correct?
Yeah, right now I am a postdoctoral researcher in Dr. Dan Caravel's lab at the University of Minnesota. And I work on a collaborative project that looks at different plantings and their benefits on both honeybees and native bees. So we have different sized pollinator plantings. So small, about an acre, and then large, so 8 acres or more. And then we have different percent agriculture around them. And so what we're trying to see is how big plantings need to be in different contexts in order to have an effect on pollinator health. So not looking so much at the interactions, but just looking at the benefits of.
So there's also a lot in the scientific literature about kind of this raging debate about honeybees out competing native bees, or not really vice versa, but there's some question about how social insects, the social colonies of honeybees, might actually kind of be disruptive to the natural environment. So is that some of what you're doing as well, of trying to look at that and where that balance is?
That's not so much the goal of this study right now as is. It might be a project that we might do.
Now he's back in here.
But it not right now, at least. Right now, it's just looking at the different benefits. Okay. But yeah, that is definitely an area of concern that we really want to stay on top of.
Yeah. And that's something too, commercial beekeepers too, when they have hundreds or thousands of colonies and moving them in and out of different areas, I think that's something that the responsible beekeepers need to realize the effect that they might be having on the local ecology. So I think it's really important for us, beekeepers, to realize, where we fit in with a lot of that as well. So I think that the research that you're doing is really great that way. You also have a kind of a good history, protracted history of doing queen research, which is obviously near and dear to our hearts here at NC State. And we've collaborated on some really great studies. But you're venturing into something new here, where you're looking at the economics of queens. So tell us a little bit about that.
Yeah, sure. So I'm really, really interested in queens. I think queen breeding is probably the most interesting part of beekeeping. And part of what the interest is as well is, the three main factors that contribute to the health of a honeybee colony is, Varroa, nutrition, and queens. So really making sure that you make correct decisions when you're replacing queens is really important for the long-term sustainability of a colony. So this is actually an idea directly from a commercial beekeeper in North Dakota. So we were playing around with some ideas, and this is really his design. His name is Zach Browning. He runs Browning Honey Company up in Jamestown, North Dakota. So what we are doing is we are going out, people from the university, me and some techs are going to go out and work directly with his crew. And what we're going to do is look at the decisions that they make in real time based on what they are seeing in the colonies. So whether they look in a colony and they judge that queen to be not going to last the year, and so they replace her, versus when they leave a queen in there. So what we're going to do is to mark down the management and these decisions that they're making, and then follow these colonies long term. So out to California, where they'll be used to pollinate almonds, and then see how strong those colonies are throughout the year and how many colonies survive. And when there are queen supersedures or replacements or colony death, then look and see like what percentage that is for each group. We're doing this with 3 different queen sources as well. Mm-hmm. So Italians, Carniolans, and then a VSH line, okay. Just out of curiosity, just to kind of keep it balanced too. But right. Yeah, so I'm pretty excited. So looking at how that benefits his operations and what decisions he's making.
So that's really great to get that kind of longitudinal aspect, and then to put it in kind of a dollars and cents measurement, which I think is really important for commercial beekeepers. But that's not the only strategy. In fact, I think that's a strategy that I think a lot of beekeepers aspire to. But I know plenty of commercial beekeepers that they just wholesale replace every single queen because of the time and effort it takes to check every single colony. They just say, well, to heck with that. We're just going to replace everybody and assume that all the queens are bad or need replacing. That must be a different cost benefit analysis too, right? And so I'm not really sure if that works or doesn't, but they're doing it, right? Is that something that you could also analyze?
Yeah, we're not going that far, but I think right. We'll get some of that with the ones that are for sure replaced. And we can compare that to the ones that aren't replaced. So we can kind of mimic that setup, but have something to compare to. And on the other end of that spectrum, some beekeepers don't find their mated queens and don't find the old queens, and then just put in queen cells sometimes, and just kind of hope those with the guard around
and assume all goes well, right? Like they don't necessarily know what happens.
And then that would be a whole other economic thing, but we're not doing those. But
right. No, that's really interesting. So I know that you've also done some of the quintessential work about how beekeepers are able to identify quote unquote poor queens. And one of the striking findings of that is that it's not really always the queen, if at all. So, describe how a lot of beekeepers go about assessing whether a queen needs to be replaced. What are they looking for? And, what may be right or wrong about that type of assessment?
Yeah, so this is a project we did with you. So what we did is there's a number of different reasons a beekeeper may replace a queen. They may be a drone layer. There might be something going on where the colony is just not growing the way the others are, and beekeepers just decide to replace the queen. Another really common way that beekeepers decide to replace the queen is based on brood pattern. So that's specifically the characteristic that we looked at within the colonies. So the symptom that we looked at within the colonies is, do colonies with poor brood patterns actually have queens that are poor? So what we did is we found colonies with poor brood patterns in commercial beekeeping operations, and then we found paired colonies with good brood patterns, and then we swapped the queens to see if it was an effect of colony or queen. And what we found is that overwhelmingly, it was not queen, it was due to the colony. Poor brood patterns tended to stay poor in that same colony, and good brood patterns tended to stay good in that same colony. So brood pattern alone isn't enough. Like, it's not a reliable way to indicate if your queen is good or not. It's not a reliable way to indicate queen quality and whether or not to replace her. It might be something else that's going on within the colony.
And in hindsight, that makes a lot of sense, right? Because the queen doesn't raise that brood. It's the workers that raise the brood, right? So it's more of a colony level trait than it is a queen level trait. But I think we just kind of get trained as beekeepers of looking at the queen's brood pattern, and that's all because of the queen. So if it's good, she's good. If it's bad, she's bad. And I think it's a lot more nuanced than that, or a lot, I should say, a lot more complicated than that. So that's something that I think is really, really helpful. And I know a lot of beekeepers are kind of rethinking the way that they're assessing their queens and to take a closer look. So
yeah, I totally agree. And then if you think about what contributes to the brood pattern itself, like at the different stages the larvae can die and like what can go wrong, you know, you can start it out with the queen for some reason laying bad, and then it would be the case that it is the queen. But there's so many different stages that can go wrong with that larval development, that egg to larva to pupa development, that you get these holes in the brood pattern. Right. You know, disease, poor nutrition, potentially pesticides in the comb, but that one's a little unsure. But yeah, so there's a lot of different things that could contribute to poor brood pattern, and that's why it's such an unreliable indicator.
Yeah. Well, I know that going back to your master's days, your main project that you did there was to look at different sampling techniques for mites. And mites obviously are kind of the number 1, 2, and 3 management problem for beekeepers. And what we've been really trying to push, especially among the smaller scale beekeepers, is always carry an alcohol wash or sugar shake jar every time you go into the colony, and every time you do, you take a sample for mites, right? Every colony, every visit, every time, like just try and get that into the culture. Not everybody does that, but that I think is what we aspire to do. But your project was looking more, again, among these larger scale beekeepers, especially the honey producers in the upper Midwest, where they just don't have time to do a sugar shake every single time they go into every single colony. They just can't do that. And so you came up with a kind of a more of a generalized sampling pattern so that they can make informed decisions. So tell us a little bit about that.
Yeah, so for my master's, what we did is we worked in 32 different apiaries, I think, commercial apiaries, and then sampled the full apiary, and then went frame by frame and sampled each individual frame for mites, and then we recorded what was on each frame. So the idea was to figure out the distribution of mites within a colony, so if they're more likely to be on brood or not, and they are, although it's not a huge significant, it might be super biological, but and then distribute the mite distribution within an entire apiary. So we used a bunch of statistics and came up with a sampling plan for both an individual colony, which is that 300 bees, and then the entire apiary, which is to sample 8 colonies in a standard commercial apiary to get the estimate of the mite population within that apiary.
Right. And so who would you recommend doing kind of the apiary level mite sampling versus the colony by colony level? Like what types of beekeepers would you recommend those two different approaches then?
Yeah, so backyard beekeepers, smaller number of colonies, sample everything. Colonies can be really different from one colony to the other. And we found that within commercial operations too. Sometimes there would just be a single colony that had really high mite levels. And, if you don't get that in your 8, you don't see that. And so in particular for smaller scale beekeepers that have fewer colonies, I think it's super important to sample each colony. Right. And then for the larger scale beekeepers, to do the larger sampling plan. So if you have an apiary, say of like 32 to 48 colonies or something, that would be more appropriate, sampling the 8. But also, if you're doing any type of breeding work, that's an incredibly important thing too, is you sample each individual colony so you can make that are potential breeders, so you can make educated choices for your breeding population.
That's a great point, right? Because the genetics of each colony is going to be different and unique. And yeah, that's a really great point. So, I know that to try to implement some sort of control measure, the percent thresholds, right, vary depending on the time of year. But those thresholds have also been going down over time, right? So it used to be, a lot higher than it is now. And so now, if you're looking at 3%, again, depending on the time of year, or more, your bees can really be in trouble. Or it used to be, a percentage of like 8 or 10% was fine, but that number is consistently beginning lower. Do you have any, or what's your take on why that is, and why those thresholds are getting kind of more and more narrow and making it more difficult for beekeepers?
Yeah, I don't know for certain by any means, but my guess would be viruses would have a lot to do with it. In particular, deformed wing virus, which perhaps has gotten more virulent, more damaging than it used to be. And so those thresholds are much lower than what they are because of the viruses themselves, which actually makes me a little more concerned about sampling too, and what it can and cannot tell you.
Right.
Because of the variability, and because if your threshold is just one mite in the spring, if you don't sample repeatedly and you get kind of it's an estimation. Okay, so you have your 300 bees, and it's representative of a colony of, say, 30,000 bees. So there's a chance that sample may not be representative of what's in that colony. And so you make a decision based on a single sample. And because those mite levels are so low, those mite thresholds are so low, I'm a little less sure about the sampling method anymore.
Yeah, no, that's a good point, right? Because when you're getting that low, each mite is a discrete number, but every sample is going to be slightly different just based on kind of randomness within the colony. And so you might get 1, you might get 2, you might get 3, right? But so really what's best is to take 5 samples and average them or something, right? But then that's a lot more time, right? It's hard to ask everybody to do that. So yeah, it's I would probably tend to agree that it's the pathogens and some of these other stressors that are interacting with the mites. It's not necessarily just about the population of mites, but these other things that mites are vectors, and that's ultimately I mean, I would love to have a situation where we just have our colonies swimming in Varroa mites, but they're healthy. I could care less then, right? But because that's not the situation that we're dealing with, and the only thing that we can do to control those viruses and other things is control the vector. That's really the only option that we have right now. So anyway, we're really fascinating stuff, Katie. Really appreciate you talking to us this evening. I think that for the last segment here, what we do is we just turn it over and have an open question and answer session. Let folks type in their questions on the YouTube channel, give some people a little bit of time to type in some questions. I'll scroll through here and see some of these here about the possibility of overfeeding packages or just colonies in general. I'll throw that over to you, Katie, because I know you've also done some nutrition research as well. What do you think about kind of when feeding too much is a potential problem?
Yeah, I mean, the main worry is that they would backfill their brood nest, and then the queen wouldn't have anywhere to lay. We generally say you can stop feeding once they have a few frames stored up of honey or nectar.
Okay.
But,
so like above the brood nest or on the side of the brood nest, or does it matter if they still have undrawn comb in the colony?
yeah, I mean, I would still feed if they had undrawn comb and there wasn't a big nectar flow going on. In general, once our nectar flow here in Minnesota hits really hard.
Yeah.
And so we can pretty much stop feeding when that hits. But if there's undrawn comb, I would just keep feeding.
Right.
until
Right.
then.
Yeah. It's also very different. You know, in Minnesota, you have winter and then you have July, right?
It's
And
no
so
left.
so everything, like the nectar flow, everything is really compact, and everything is very here, right? We have 3 months of a non-broodless winter for the most part, at least in the lower elevations in North Carolina. And so there's months where there's nothing out there at all, and so it's a very different dynamic, I think, sometimes depending on where you are and what your immediate local ecology is, because things can turn off like a tap in the heat of the summer, and the bees can really struggle during that period. So but yeah, you definitely don't want to overfeed. Anybody else want to comment about overfeeding or just stimulative feeding?
I'll throw in a comment about the small hive beetles. I don't like having
Oh,
frames that are not being protected by the bees.
that's a very important point, especially when it comes to supplemental feeding of protein and pollen patties. But that's even true with syrup, right? They'll reproduce in the syrup too, right, Jen?
Mhm.
Yeah, that's
And on the brood.
yeah, and on the brood. Yeah, once you get the outbreak, then it's hard to contain them. No, that's a good point. So here's another question from Cynthia here. What's the summer threshold level for Varroa in the southern region down here? So midsummer, what's the recommended threshold? I believe it's 2 or 3, isn't it, percent?
Yeah, I would say the same thing.
Is that the same threshold up your? Okay, so Lewis says it's 3%. I think it's 5% in the fall, but I still think that might even be still a bit too high. This time of year, it's closer to like 1 or 2%. So it really does depend on the time of year and how much time the mite population has to build up. But yeah, so for midsummer, 3% is a good number where you really need to step in and start doing some control measures. Sorry, Lewis, go ahead.
I was going to say, I think Katie did some really good work on what you can expect if your mite levels are above 3% in June, right?
Yep.
Yeah. Odds of the colony being much smaller later or dying are much higher if it's above 1% in June.
Yeah.
So that really underscores the importance of monitoring during the season. I know a lot of beekeepers don't monitor if they monitor in August or September, and
might be too late
at that time. If the mites are out of control, it's really too late. So it really is important to make monitoring part of your all season management plan.
So that brings up another study of yours, Katie, that I think is always really great, because I get this a lot from beekeepers when they say, well, I don't have mites because I don't see them. Right? Or I don't see the crimpled wings. I don't see evidence of them, right? But if you go and you do an alcohol wash on their bees, they're going to be already over threshold, right? So some of your studies really said, once you start seeing the mites, they're already in dire straits, right?
Yeah, once you start seeing either mites on adult bees, because most of them are underneath on the abdomen where you can't see them, and so we always take pictures when they're on their backs, which is not where they normally are. Yep. Once you start seeing that, once you start seeing the foreign wing virus or any signs in the brood or parasitic mite syndrome, they're already over threshold. The odds are, at least. So yeah.
So that just underscores what Lewis was just saying, which is you want that monitoring plan, because by the time you see the evidence of the mites, they're already in need of help. So you want to be proactive and preventative in that. So yeah. All right, let's see any other questions here. If you treat for Varroa mites now, when can you extract honey? I'll open that up to anybody, not just keep putting Katie on the spot here. But
Depends on what you're using.
yeah, so Don, why don't you describe some of that? If, like, let's say you come across a colony that's above threshold right now, or just, has kind of worrisome mite levels right now, but you want to harvest your honey that they're kind of collecting and everything right now. What are some of the options that you would recommend to beekeepers who want the best of both those worlds?
I wouldn't. Well, formic is the only one that you legally can.
So that's formic acid, which is an organic
Yeah.
biopesticide, right?
Yeah, but if you've got a single colony, which we're generally talking about, if we're talking about Napier, it's another situation.
Right.
But if you've got a colony or two, it might be better to put those supers on another colony and hit them with something like formic acid and then put them back on. You could leave them in there, but I don't know. I just don't feel comfortable
Yeah.
doing that.
Yeah, well, again, this is a lot of the problems that the mites introduce, is that sometimes you have to make these really hard decisions. It's either control the mites and keep the bees healthy, or your honey crop. That's not a great choice. It is very hard to kind of have your cake and eat it too. Lewis?
I was going to say, again, this underscores the importance of early season monitoring.
Yeah,
if you monitor at population buildup in February or March, you have time to react,
that's right,
get things cleaned up before you have to put honey supers on.
Yeah.
So I think it's all about being proactive and not reactive.
Exactly
That's really
right.
the key to success, I believe.
Yeah, exactly right. That was a great question. Thank you for that. How does the queen cell get populated? So do the workers move an egg into the queen cup, or what happens? So anybody want to take that? Go ahead, Don.
I can think of two people who I respect who say they can move eggs. And even though I respect both of them, one of them is no longer with us. No.
Yeah,
Now,
just because it may
one-
be physically possible and it's been observed once in a blue moon doesn't
Yeah.
mean that that's how it's always done, right? So how is it normally done, shall we say? How do the eggs get into those swarm cells?
The queen puts them there.
Correct. Yep. That's by and large usually what it is, and again, that's in response to overcrowding.
One of the other people that is still alive, I guess he's still in British Columbia, and he's a very knowledgeable beekeeping researcher. But I think he must have seen something happen that was an anomaly.
Yeah, that's always my take on it too, because otherwise we would see eggs in queen cups above excluders all the time.
Yeah.
And
he did observe that.
He did observe that, yeah. And I'm not denying that, but that might have been a rare event.
I think he probably didn't observe a little kink in the excluder or something.
That's an alternative explanation as well too. Yeah. But usually they're doing it on purpose, right? Because they're wanting to swarm. But of course, that's different and distinct about when a colony wants to simply replace an old queen. We don't really think that superseded queens lay the eggs in the cups. Those are where the workers will modify a worker cell, start feeding it royal jelly, and make queens that way rather than the queen laying the egg. So it does depend on the context of why the colony is raising the queen.
Yeah.
Well, all great questions as usual, everybody. Sorry we couldn't get to all of them here, but I do want to wrap things up here by reminding you that we're doing these apiculture onlines every last Wednesday of the month. So that means the next one that we're going to be doing is May 26th. So again, at 7 p.m. The timely topic this time is going to be presented by our guests of honor and interviewees, Ann Spafford and Denisha Seth Carley, both here at NC State in the horticulture department. And it's going to be an extremely popular and very important topic that is on bee plants and on planting pollinator gardens. They both just published a really excellent book that describes all sorts of different bee plants and garden designs. And so pretty much the entire hour is going to be addressing this really, really excellent issue. So be sure to tune in then. So until then, thanks again for joining us. Again, be sure to subscribe, like, comment on our YouTube channel, and hopefully we'll see you next month. Thanks again.